Welcome to my YouTube channel Fast Track Learning. In this video, we will understand the science behind CDK inhibitors. We will learn about different types of CDK inhibitors, their mechanism of action, and benefits they offer in targeting cancer cells. CDK inhibitors will help us in cancer treatments, so we have to pay attention to CDK inhibitors. So what are CDKs? CDKs are family of protein kinases that play a crucial role in regulating cell cycle. CDKs stand for cyclin-dependent kinases. First of all, I will help you to understand cyclin and then kinases. Here you can see cyclin. Cyclin makes a complex with CDK. So, cyclin dependent means CDK depend on cyclin. It means CDK needs cyclin, so cyclin bind with CDK and make a complex. Cyclin makes CDK active to perform its function. That is why CDK need cyclin. Second concept is kinases. CDKs are kinases. CDKs are family of protein kinases. What are kinases? Kinases are those proteins that add phosphate group to amino acid in a protein. So we understand two concepts which are very important concepts here. What are these? These are CDKs depend on cyclin. CDK cyclin complex is very important because CDK needs cyclin. And second important concept is they are kinases. It means they phosphorylate a protein. Then we have CDKs. I will share you their names. What are the names of CDKs? We have three CDKs: CDK46, CDK2, CDC2. Here we have cyclin and CDK complex. CDK can be any of these: CDK46, CDK2, CDC2. Now I'm going to tell you you see in this complex we have CDK and we have cyclin. Now I'm going to tell you what are cyclins. Actually, cyclins are also very important. CDKs require binding to a cyclin, which is a regulatory protein, to become active. We have cyclins, cyclin A, B, D, and E. So here cyclin can be A, cyclin can be B, D cyclin, or E cyclin. Cyclins are regulatory proteins that bind with CDKs. So here we understand two very important concepts. You have to memorize cyclins are regulatory proteins. They bind with CDKs. So and also you remember the names of these cyclins, which are A, B, D, and E. Now I'm going to tell you one thing that specific cyclin will bind with specific CDK in a phase of cell cycle. Like D-type cyclin will bind with CDK4/6. Now we are going to understand role of cyclin CDK complex. We have cyclin bind with CDK. Cyclin and CDK combine to make a complex which is known as cyclin CDK complex. So cyclin will activate CDK and CDK will phosphorylate a target protein. Here we see phosphorylated target protein. So important function will be performed when cyclin CDK complex will form. First step is cyclin CDK complex formation, while the second step is phosphorylation of target protein. When the target protein is phosphorylated, so this is very important for the cell because it will allow to pass through different phases of cell cycle. As a result, different functions can be performed. But in case of excessive proliferation, we need CDK inhibitors that will inhibit the cell cycle. So, what are CDK inhibitors? That inhibit CDKs. I'm going to tell you that there are 7 CDK inhibitors. What are their names? These are P16, P15, P18, P19. P16 is written first, P15 is second because the sequence is INC4A, INC4B, INC4C, INC4D. That is why it's easy for you to memorize in this sequence. If you memorize P16 first and then P15, so INC4A, then INC4B, then INC4C, and then INC4D. And the other three are P27 KIP1, P57 KIP2, and P21 SIP1. So these are 7 CDK inhibitors that inhibit CDKs because they have very important role to prevent cancer. Now I'm going to show you a small molecule inhibitor which is a synthetic drug. What is its name? Its name is palbociclib. This drug inhibits CDK4/6. This drug is inhibitor of CDK4/6. CDK4/6 is very important when it combined with D-type cyclin. So this is very important for transitioning from G1 to S phase, because in S phase DNA synthesis will takes place, but when this synthetic drug will inhibit CDK4/6, so it will inhibit cell cycle, it will halt cell cycle. So you can understand from this concept. Now we have CDK inhibitors mechanism of action. First of all, CDK inhibitor will bind with CDK, with the active site of CDK. When CDK inhibitor is bind with CDK, so CDK cannot bind with cyclin. When CDK cannot bind with cyclin, cyclin CDK complex cannot be formed. So cyclin cannot activate CDK. When CDK is not activated, it cannot phosphorylate target protein. So no phosphorylation of target protein. As a result, what will happen? As a result, it lead to cell cycle arrest. It means cell cycle will stop because no phosphorylation of target protein, so cell cycle will stop, it cannot take place further. So cell have two advantages. It can perform DNA repair because there was a DNA damage. That is why we want to arrest cell cycle so that DNA repair can take place. Also, if there is damage is too much, there is also another option which is apoptosis, which is programmed cell death. By these two processes, it will stop proliferation of cancer cells. They help to stop proliferation of cancer cells, making them a valuable tool in targeting cancer treatment. Now, I'm going to tell you the conclusion. Understanding the molecular biology of CDK inhibitors is essential for grasping their role in cancer treatments and cell cycle regulation. If you have any specific aspect you would like to explore further or need help with video content, you are feel free to ask. Also, please like, share and subscribe this channel. I will make further valuable content for you.